Okay, hello. I'm happy you came for the last lecture of this wonderful week. It's a challenge for me because all the speakers have been so great up to now. I hope I don't disappoint you. So there is I will somehow use, or will go on with what David was doing. But you see, I don't have chemical reaction networks, I have biochemical reaction networks. Biochemical reaction networks are chemical reaction networks, but not of any kind. Okay? So as David said, he mentioned this Harsh and Toll's result characterizing which system of polynomial equations come from as a right-hand side of one of these ODE systems. But I don't want to work with very general because these are too general. Nothing interesting can be said about this. They are way too general. So I want to focus on some chemical reaction networks that are relevant to biology. Okay? And so this is what I'm going to talk about. And so this is a very rough plan. I first start with a small, revise slightly this, what's a chemical reaction network and which is the main goal here. And then I will speak a little bit about multistationary and persistence. And then I will show you some important biological networks that in principle look quite different. And then I will show you that they all share a structure that we call a messy system. And if you feel that this is close to Leo messy, it is on purpose. And then I will roughly talk about other approaches to multistationarity, and then I will end. Okay, now the other side. So this is one example. This is similar to the ones that David was showing to you, except that now the variables are not A, B, C, D. The way that chemists and biologists call the variables is the true name of the chemical species. In general, chemical species here are in this setting are huge molecules. It's not like oxygen or nitrogen, is a huge protein. Okay? And they put the names of these proteins because this gives sense to what they want to do. Okay? If you put X1, X2, X3, they all look the same, but like this, they know that here this is what's called a two-component system. At first I hated this, but then I got used because it's much better because it relates to something, how this component is acting, how the other is acting, which ones can interact, which cannot. It's much useful. If you want to put it in a computer or do calculations, you have to number them. But it's really useful for me to keep them like this. I won't in the rest of the talk, sorry.
So this one component is the HK stuff and the other is the RR stuff?
Yes. So you see here, so here it's called, there's this kinase, this is an enzyme, it's called hybrid histidine, I don't know how to pronounce kinase. And it has what, in general, these proteins have what's called a domain where there can be phosphorylated. This means that there is a phosphate group that can be added there. And when the phosphate group is there, in general, they say this is activated. Activated means that now it can go and participate in another reaction. Okay, so in general, it's the active state where it can start a signal transduction process. Okay, and so Fischer and Krebs got the Nobel Prize in physiology and medicine because also there are some proteins that put phosphate group and some others that take it away. And this seems to be like a futile thing to do. But this is the way in which the energy is, one of the ways in which the energy is transported. So here you have this, and it has two sides. So the phosphate route can go to the first one or the second one. See whether it is a 0 or a P, it means whether the phosphate is there or not. And this is the directed graph. And then there is this RR, it's called the response regulator protein, which can be unphosphorylated or phosphorylated. Okay? Next transparency will have X1, X2, X6. But I put different colors because you see that they have different roles. Okay? Okay. So now it's here. So now it's in more mathematical form. And from this directed graph, using mass action kinetics, the K1 up to K6 are positive real numbers, or we can think that there are parameters, and they live in R positive to the 6. And these are the the ODE system that comes from there. So here what happens is, in general, this capital X is one chemical species. Small letter means the concentration of the species. Really, our variables are the concentrations of the species that we assume that evolve in time. And they evolve in time according to this system of ODEs. Okay? In general, capital letters is oxygen, and small o, is the concentration of oxygen, which is kind of a probability, the probability of having a molecule of oxygen there. Okay? Here. Okay, here are the equations again, and it is easy to see that in this system of equations, it's easy to see that F1 plus F4 is equal to 0, and also F5 plus F6 is equal to 0. This means that the sum of the derivatives of x1 plus x2 plus x3 plus x4 is 0. This means that for any t, this sum takes a constant value that I'm calling T1 and T2 on the other side. These T1 and T2 are called total amounts or total concentration constants. So in fact, we have 6 variables, this concentration of the 6 species, but the trajectories are constrained to a 4D plane because it has 2 equations in 6-dimensional space. So the trajectories happen in this 4D plane cut out. If these values, T1 and T2, depend on the initial condition or any point in the trajectory. Once you have the point, you know where you cannot go away from that, from this parallel translate of the subspace, which is defined by sum of the xi's equal to 0, 0. Right? This is called the stoichiometric subspace. I forget. Okay, so this is more or less what David has written. In general, one has a directed graph which is labeled. On one side, there are the labels on the arrows, which are the reaction rate constants, and also there are labels on the vertices that correspond to the complexes. In fact, what we are going to do is, if we have x2 plus x6, this will give a monomial, which will be x2 times x6, because this is going to be the exponent. This is what he was calling the complex. And a complex can be seen as a linear combination of species, but in mass action kinetics, what we are going to do is look at the monomial, that comes taking x to the y. And this y is, for instance, it should be E2 plus E6. So in this setting here, in this example, there are 6 arrows. So there are 6 reactions, 10 complexes, 10 nodes, and 6 species, x1 up to x6. And from this combinatorial information and labeling, you get this ODE system that I have just shown you, which is again here. And the mass action kinetics is like this. So here, dx with respect to t means this is a column vector, dx1 with respect to t, dx2 to respect to t all the way up. And so this has to be interpreted as also as a vector. Here, the kij's are, E are the level, is the name for the arrows in this directed graph. Kij are positive numbers. X to the yi are monomials, and and the xj minus y, yj minus yi is the difference of these 2 vectors, exponent vectors. So this difference is an integer vector. Okay. So the vector goes like here, and the differential goes like there. And this is what you get. Okay, now I'm very happy to be able to tell you a little bit of history that David has not told you, and I found fascinating. So this story, the mass action kinetics, maybe you already perceived during his lecture, is very mathematical, this modeling, right? Didn't you feel that it is very mathematical? In fact, it was proposed by a chemist together with a mathematician. And they were in Cato Gulberi, and was a chemist, and Peter Vage, I don't know how to pronounce it because it's Norwegian. And they were very good friends and fantastic people. If you go to Wikipedia and look for their biographies, they did very interesting things, both of them. And they were very close friends, and in fact, this, Vage's second wife was Gulberi's sister, and they are Ragni Pine's great-grandparents. You probably know her. She's a well-known algebraic geometer. She also worked in applied algebraic geometry, and she pointed to me that they were relatives. What happens with their work, which is now more interesting to us, is that this was published in Norwegian in a journal in 1862, but obviously it was not acknowledged. It was not read. Then they wrote the paper, a version in French in 1867, but still at that moment lingua franca was German. And in the process, someone, Van Hoft, a Dutch scientist, found the same modeling, but eventually they published their work in German, and everyone acknowledged it, that they had proposed this modeling. Okay, I don't know why Wage appears here. Okay, the first observation that you have already noticed is that what we get on the right-hand side are polynomials with real coefficients. They carry the combinatorial structure of the directed graph, but we have polynomials. This is a polynomial ODE system, and linear relations among the vectors give rise to linear conservation relations, these equations that are, there are other conservation relations that come from some other sources, but in general, the ones that you get come from the linear relations among these exponent vectors. And this is the stoichiometry. And by the form of the equations, I'm not proving this, but it's true that if you are supposed to be modeling chemical species, if you start at a point in the positive orthant, then no species can be extinct. It can tend to zero, but it can never, no species can take the value zero. So you will see, I'm not defining steady state, but let's define them now. Steady states are the values which make this equal to zero, where the derivatives are zero, okay? If you are at a steady state, these are the constant trajectories. You don't move because the derivative is zero. So you never go to either you are at a steady state or you never reach it, okay? So what happens is that the trajectories that converge, they don't need to converge, but trajectories that converge tend to a steady state. So understanding steady state tell you things about the trajectories. But it's not that if you start in the positive orthant, you will be in the positive orthant forever. Okay? Also the non-negative orthant, this is said that the orthant is forward invariant for the dynamics. Or it also happened with the closed orthant. And in general, these rate constants are unknown, and they are difficult to measure, or even there are some results that say sometimes they are impossible to be determined. There's a paper by George Creation and Krakasian Pantea, there's some other ones. Okay, so the very general goal, what we would like to do as mathematicians is, given that we have a mathematical model, that they decide when this makes sense, not us. So we would, in general, these are built using the biologist's intuitions and experiments. We would like to give them a black box which spits out some properties of these networks, okay, from the structure. So here is the definition of the steady state. It's a non-negative point. For me, in the rest of the lecture, I will concentrate in strictly positive state, steady state that lie in a positive orthant. For which all derivatives vanish, so the zeros of this polynomial system, real polynomial system. And for instance, you saw some pictures that we can look at. So this is the positive orthant, and we look at the zero sets of the Fs, like this, and then we have this stoichiometric compatibility, so we have these subspaces where they are supposed to be parallel, okay? These parallel, you have this linear space, and as you move the t's, you're going to have this family of linear subspace in affine varieties. And then it might happen that it hits the steady state variety in 1 point, or maybe you get more than 1 point. If you get more than 1 point, we say that the system, for this particular choice of rate constants and total values, the system has to be multistationary. In general, what we say that the network is multistationary, or has the capacity for multistationarity for some choice of k's and t's. You can get that more than one positive steady state in the same, what's called the same stoichiometric compatibility class. This is the name. What did I do with the wonderful? I had it right. I see it. Okay. Here maybe it's the same picture that is easier to understand. And for some values, like here, you can have only one, but the double one. Here's where the intersection is tangent. Okay, this will be a degenerate steady state, and the other ones are non-degenerate steady states. So if we were in the complex domain, if these linear subspaces were general, the intersection will have degree many points. But on one side, they are not generic. There are some fixed spaces in some direction that might not be generic, but also we are interested in the real positive solutions, not the complex solutions, which make things complicated. Okay, so this is the usual multistationarity pictures that you find in the literature. What is not said here in general is, as you saw, for instance, in the example that I showed to you, which is really, really tiny with 6 variables, the picture should be in R6. More than that, there were, I don't know, many K's and T's, so the picture should be in the space of K's and T's and X. It's a huge space. So when they make these pictures, they have made many choices. They have fixed the value K1 equals 1, K2, 0.5. This is just a section of this huge dimensional thing, but it's never really very clear how they pick the section, and in general, they do massive simulations. Okay? Massive simulations to find these pictures. Okay, but in general what one expects is picture like this one upstairs. So there is a region which is called the monostationary region. In general there are only 2 parameters that they allow to be free. So when in this region there is only one positive steady state. Then you have this blue region, which is called the bistability region, which means that in general you have 3. 2 of them are stable steady states, which are not defined in none. And then you get also 1. In general, the regions where you find many positive solutions are tiny regions. Okay? Okay. And so why is this interesting? So this is, I don't really know biology, but it's, in general, you maybe heard this, that most cells in our body have the same genetic information. Okay? But some of them develop as a brain cell or skin cell or liver cell. And in principle, they say that this multistationarity property is that allows the cell to take decisions, where it's going to approach this steady state or the other steady state. Okay? I don't know, this is just, for me, it's a very vague idea, but it's really an important concept. Also, this guy, Ferrer, he visited Buenos Aires. He was invited by the physics colleagues. And he has, I guess, like 2 PhD degrees. One is in biochemistry, and maybe the other one is in physics or medicine, but he likes math a lot. He had a major in math. So it's very nice to talk to him. And he has a fantastic article where he says, it's 09, but I think probably it's similar now, that the current state of system biology is like planetary astronomy science before Kepler and Newton. He says that cannot be studied without math and physics. So maybe there is some room for us to say things and also to isolate in small networks or small group of things the complex behaviors. Okay, this is the hope. We'll see. And also something similar is said in this paper by Thomas Soule, is a mathematician working in arithmetic, algebraic geometry, K-theory, but he also has a couple of papers on that. So one important property is persistence. So persistence is the following. So if I start here and a point here, okay, it might happen that I didn't, not in my picture, but there could be boundary steady states. There could be solutions of the Fs that lie on the boundary. As I told you, if a trajectory converges, it has to converge to a steady state. Okay? So if I start in this line and my trajectory converges, if this is the initial point, I could maybe go here, maybe here I go there, and here no, but I could also, if there was a steady state here, I could go there. Okay? It might happen. It's not that I'm going to be, in fact, it's not, you are not going to be like this. It's what's called omega limit point, so the trajectory can wander around, but there is a sequence of points in the trajectory tending to this boundary steady state. So in this case, the trajectory will get as close to the boundary as you wish. This is a system which is not persistent. A system which is persistent is a system which it's away from the boundary. How much away depends on where you started, but this is a system which is persistent. Okay. This persistent means that those species which is present can tend to be eliminated. Can never be zero, but can tend to be eliminated in the course of the reaction. So I will mainly deal with these dephosphorylation futile cycles, or not versions of these. So in general, you will have six species in this small picture, 6 complexes, which are the nodes. The species are are S0, S1, and they have different names because they are called substrates. These big names, S0e and S1f, they are just one variable, one species. I could call them Y1, Y2, or X1, X2. But they keep this name to understand that this S0e is one species that comes when S0 binds to E. And E is generally an enzyme, so I have a substrate, S0, E is an enzyme. So there is a reversible reaction that gives this intermediate species, and then you get S1, which in general I assume that this is S0 phosphorylated, and E goes away. This is an enzyme. It produces the reaction, catalyzes the reaction, and goes away. And there is another enzyme, E is called a kinase, and F is a phosphatase, that takes the phosphate away. Okay, all this picture with these 6 Ks and all of that, we will represent this shorter way. Okay? When we see a picture like this, it means like that. And so this is similar, but I have a 2-sequential phosphorylation. There are 2 sites which can be phosphorylated, and then E comes again and gives a second phosphate. So I put different colors. You see there are S0, S1, and S2. The blue ones are intermediate and monomolecular complexes. And E is one enzyme, and E is the other. Okay? And then we are going to call the concentration with small letters, but I will keep the colors, hopefully. And here are the equations read from this network, and these are the conservation relations. Okay? Which there is a theorem that explains that this can be also read from that network, from that structure. Also, here is another, I will show you some other important example of networks. So here, this is a phosphorylation cascade. What happens here, otherwise I will make a mess. What happens here is that S1, which is the phosphorylated substrate at the first level, is the enzyme at the next level. So you produce an enzyme, one is phosphorylated, it's an enzyme in the other one, and all the way like this. In general, this is what happened. The first reaction takes place at the membrane of the nucleus, of the cell, and then it starts something inside. Okay? This chain of signaling reaction. So as you see, each of these small pieces has 6 Ks, 6 variables. There are many variables here. Okay? In this picture. And here is another one, and here you have the true name or acronyms of the molecules that are involved. And here is called a retroactivity because there is this, the last product there acts as an enzyme upstairs. Okay? And then this one, this is a small cascade, only 2 layers. And here again, the product with S1 is going to act as an enzyme in the second layer, except that the difference between these two is upstairs, it is assumed that there are 2 different enzymes that take the phosphate away. And downstairs there is a single one, this F in red. You see here, upstairs there is F1, F2, and here there is an F. And in fact what happens is they behave very differently. The first one is monostationary always. It cannot have more than one positive steady state in one class, but this one can. Okay? This has the capacity for multistationarity. This was first proved in a paper by Elisenda Feliu and Karsten Bjoff. And also this system that was studied in this paper by Karsten Conradi and Xu is a processive phosphorylation. So you have intermediates until all the way you go to S0 to Sn, which has the N phosphates all of a sudden. And then these are small motifs that you can find in this paper by Elisenda and Karsten. And Uri Allen, he wrote a book. He's originally a physicist, and then he worked in biology. He also does theater. But he has a very well-known book. They talk about motifs that are small motifs that occur much more frequently than they would occur just by chance. And so they say, well, they probably have a meaning or an importance. Let's study. The only thing that we can do is study small motifs. But there is a good justification for this. Okay. And this is another network that was proposed or studied by Stas Schwarzman, which is a very interesting scientist at MIT. And so what we are going to show you just very roughly is that we identified with my former student, Mercedes Perez-Millán, we identified the common structure in all these enzymatic mechanisms that I have just shown you. And this is an explanation of the name MESI. They describe modifications of type enzyme substrate of swap with intermediates, and which allows us to prove general theorems for all these mechanisms at the same time. Okay? All the previous ones are MESI systems. So now we have a less general goal. We are not going to start with anything. We're going to start with a MESI network and then try to properties, okay, without having the constants. Okay, what's the definition? The definition takes 1 page, but it's an easy definition for us. The main property of the MESI system is that there is a partition of the set of species. Okay? As you saw, the world is red, blue, green, because they have different functions. Okay? So, and for the chemists and biochemists, it's obvious that they are different. This is a substrate, this is an enzyme, this is whatever. So there is the main point is that there is a partition of this as a disjoint. You can write the set of species as a disjoint union of one of these subsets, which could be empty, is called the intermediates. That we had intermediates in some of the examples that I showed to you. And then the other ones are non-intermediate of core complexes. This is just a terminology. But they have different properties. And so there are 2 types of complexes: intermediates, which consist of a single intermediate species, they are monomolecular, or non-intermediates, which could have either 1 or 2. But if there are 2, they have to belong to different subsets. Okay? I cannot have 2A. Could be A plus B, but A has to be in one subset and B in a different one. I cannot have A plus A. And I need a notation. This notation, this is the arrow with the circle, means that you can go from here to there via a path of intermediates. You cannot hit any core complex in the middle, but you can have through a dozen or no matter how many intermediates. If you can do that, we put this arrow with the circle there. Okay. So here what you see, let me go kind of quickly. But in fact, this is a set of axioms. Okay. This is they prescribe which is the set of allowed reactions. Okay. And this, I'm not going to read them for you now, but there is a set of allowed reactions that should satisfy all these properties. And what is interesting to me is that it seems to be kind of restrictive, but most popular biochemical networks do have this structure. Okay. So all the complexes have either 1 or 2. If I go from 1 to 2, 1 is an intermediate. And also intermediate has to be intermediate. Intermediate means that there is at least one reaction coming from a core complex through intermediates to it, and it has to eventually react to another core. It has to be through intermediates in the middle of 2 core complexes. And also maybe let's look at this one there. So if via intermediates I go from this to that, then these and these belong to some S alpha and these to a different S beta, but they have to belong to the same. If I have alpha on one side, I need to have it on the other one, etc. Okay, I don't expect you to understand this now, but it's not very complicated, it's one all observed. And this was inspired by several previous work. In fact, the first work is a very nice article by Thompson and Jeremy Gunawardena, which is called the rational parametrization of the geometry of the steady-state variety, because there is an underlying linear structure framework behind that. I think Jeremy was the first one to realize this clearly. And also there's a couple of nice papers by Jill Nyakaja in the Journal of Mathematical Chemistry. But we think that we generalize all them in a nicer way. But of course, we could do this because they had already done that. And just a small terminology, if we go xi plus xl to xj plus xl, so xl is at the end the same as at the beginning, then, so this could be through intermediates. Then we say that XL acts as an enzyme, but it could happen that the phosphate group is transferred in what we call a swap. It could be you have A and B is phosphorylated, and you go to A gets phosphorylated and B does not. Okay? It could be no enzyme, or both of them are transformed here. And here in my structure, the A's would be in one subset and the B's is going to be in the other subset. This is a swap. It's like if the phosphate jumps from A to the A's, from the A's to the B. This also happens in many examples. But these other works had the restriction: if something is a substrate, it cannot be an enzyme, and we allow it to be an enzyme in a further to allow for these cascades. And when we have a messy system, then it is kind of easy to find linear conservation relations which have coefficients equal to 0, 1. So we add over all the variables in one of these subsets in the partition. And here we add all the intermediates that we hit going from one of these core complexes. And the whole sum has to be constant. You see here that the coefficients of this linear form are 0, 1. And in what we can read from here is that any complex is in one of these, because we have a full partition of the set of species, which means that if you add all these equations, you will have something with all linear form with all positive coefficients, which has to be constant. If you have something like this means equal to a positive constant. This means that all trajectories are bounded, that all the concentrations are bounded. Assume that let me stupid thing. If I have x1 plus x2 has to be some positive constant, and x2 plus x3 has to be some positive, it's not exactly the shape. Then I have x1 plus 2x2 plus x3 has to be some constant, which is a positive real number. So this means this is a bound for this concentration at steady state, all the way, in particular, say, for this concentration, and this divided by 2 as a bound for this, and all this is also a bound for that. So the trajectories are going to happen in a compact, all this. So these places, in fact, the dynamics does not take place in these affine subspaces, which are translate parallels of some subspace, but we need to intersect it with the positive ortho. And in this case, this intersection is going to be compact. Okay? These systems are called conservative. So all of them are conservative. And this theorem implies, I said, that all of them are conservative. The solutions are the, and this implies, using a result of the ODEs, that then the trajectories are defined for any positive time. In general, you can start, and you can take an ODE system that looks like this one, even in one variable, and the solution could be 1 over x minus 1. You start at 0, when you get to x equal 1, you go to infinity. Okay? But because of these conditions, trajectories are defined from 0 up to plus infinity. So it makes sense to take the limit in plus infinity, etc. Okay? Which is also hidden in everything, but it's never, very few times is spoken aloud. Okay, so one question is when these linear relations that I'm showing there span all the linear conservation laws, and we give different restrictive conditions, but most common biochemical enzymatic models do satisfy them. They are in general combinatorial conditions on the network, and we also give counterexamples. If this is not satisfied, then it could be more. And then this is something that we took from a paper that I love from Elisenda and Karsten Viuf, which is called Simplifying Biochemical Networks. And so these pictures are from their paper, so they are a little bit different. So we assume that we have these three core complexes, and these capital Z's are the intermediates. Okay? Here, there are three different ones. Then what we do, we are going to get rid of the intermediates. Algebraically, we are going to put their equations equal to 0, eliminate, and substitute. But what we are going to, from these three, we are going to land into the same one. What we are going to do is y1 goes to y2 via this intermediate, so I draw this arrow. And here it goes there, so I draw this arrow again. So in all the three cases, there is a way. For instance, I can go from y1 to y3 this way, but there is a way, so I draw this. From any of the three, I get the same graph on the left, where the intermediates have been eliminated. And in fact, what one can do is one can then change the rate constants. So algebraically, one solves for one and replacing the other one. And then we have to change the rate constant, but this is well explained in their paper, how you do that, and you get the same equations that also come from some mass action kinetic networks, but really the dynamics is different, but the algebraic equations defining the steady states are the same. So and we get a new network. Here, let's say that the constants are called K1, K something, and here the constants are some rational functions on this case, but they are explicit, and the denominator are some of positive terms, so they are well defined over the positive order. So there is a way to somehow get rid of these intermediates, and we get another associated graph called G1. And okay, this is, and from this we construct another graph. So here, this is the doubly phosphorylation. So the intermediates here are called U1, U2, U3, U4. We take them away, we get this, but we get new rate constant. This is an important mechanism in bacteria. I think it's called MZ on PR, which satisfies an unexpected property, which is called ACR, that the value of one of these is, so the steady state variety is infinite, but even if it is infinite, the value of one of the coordinates is always the same. It does not change as you move. It's constant whatsoever. Okay? So this means they say plain that this is a way to regulate, that this is very important for this bacteria to have this concentration fixed, and this mechanism allows you for this. Independently of the total amounts, it depends on the case, but it's independent of the total amounts, the concentration of one species is the same. So this is an important mechanism because of that. And here I do the same. Here there are, sorry, this is not what I meant. here there are these two intermediates, and here I have eliminated intermediates. And then there is a new graph, which is called G2, G2 circ, that I'm not going to explain now how you get, but it's easy to get from here. There is a way of getting another graph which allows to describe the steady state pretty the same way. And the conditions for this graph to be nice are also combinatorial on the given graph. And so the first condition is how can we, I forgot to explain one graph. Okay, so there is one graph, there is another graph, which is this GE. So in this picture, there are the partition of the species is one, there are the intermediates, which I'm not showing here. Then one set is S0, S1, another one is P0, P1, and then there is E and there is F. So here I have a graph where the nodes are these subsets in my partition, the core subsets. And then I will going to put an arrow if any of the species here acts as an enzyme for a reaction between species there. So here F adds an enzyme for the S's and for the P's, so there are these two arrows, and E is an enzyme here, and S1 is an enzyme there. And then we look at this graph, and we see this graph does not have any directed cycle. This tells, with a further condition, that there cannot be boundary steady state. So if there is no directed cycle in this graph GE, then G has no boundary. And I think we need to ask maybe some other hypothesis that I maybe forgot. G has no boundary steady state in any positive stoichiometric compatibility class, so the network is persistent. And it's a very easy graphical condition. And here, in this case of this bacterial mechanism, in fact, this is the graph GE, because E acts on, there are two groups, the X's and the Y's, and one is each one acts as an enzyme on the other one, so we get this reversible thing, which is a cycle, and indeed there are boundary steady states here. Okay. Now, this is one theorem, which is one in a long list of theorems, very long list of theorems, that go back probably to work of Feinberg, for sure, and probably to this work by George Gresham and Marty Feinberg that David was mentioning. And then we distill this theorem in a paper with, I call the 1,000 authors, maybe there are 7 or 8. One is Anne, one is Elisenda Carsten, all the suspicions are there. We met in Dagstuhl, which is like overall for computer science, and we all felt that we were all dealing with some sign conditions for multistationarity, and so we assembled all this together. And then with Mercedes, we clean this, which is easier to clean, but after all this work, in case the steady state variety can be described by binomials. In fact, we also give conditions on the graph under which this can be done. We give conditions under which the good thing is that if it can be described by binomials, it can be parametrized by monomials. And then there are other instances in which you can rationally parameterize your varieties. So these are not general algebraic varieties, because a general algebraic variety cannot be parameterized. But very frequently in this setting, varieties can be parameterized. And we also give conditions and explain many things about this. We call them S-toric Messi system, meaning structurally toric. But I won't define this for you now. But just let me give you the flavor of the results, okay? So we assume that it is cut out by binomials. For instance, let's assume that we have x1 up to x4, and one equation is x1 squared minus some some constant mu of k, x2, x3, and our favorite example, okay, x2 squared minus mu prime k, I don't know, x1, x4. So these equations describe the positive steady states. They need not be even in the ideal of the S, but they just describe the positive steady state. In these examples, they are really in the ideal, and under some further hypothesis, we can get them just operating linearly. Okay? Linearly, I mean over R, not in the polynomial ring. And all this is based in this matrix 3 theorem that David was mentioning. So from this, we associate two vectors here. The exponent will be 2, 0, 0, 0, minus 0, 1, 1, 0. So we write this 2, minus 1, 0. And similar for the other one. For each binomial, we get an integer vector, and we form a matrix B. I put in a matrix B, I take all the binomials, but just keep linearly independent ones. They are enough to describe the positive solutions. Or it can be parametrized by monomials in a dual matrix. So A and B are Gale dual matrices in a dual matrix A. And then we have this subspace, the stoichiometric subspace, which is generated by the difference in the complexes each time there is a reaction. And these S perp are the equations. These are the ones that have like x1 plus x2 plus x3 plus x4 equals t1, x5 plus x6. These are the equations of S. In fact, the equations of S are equal to 0. So I have S and I have S perp, which are the equations. These are generators, but I can also give it by equations. And if it happens that the rank of these, so I put in a matrix a system of generators of the annihilator of S. If this rank equals T, and we also give conditions for this to happen, if this happens, then the conditions are the following. Let me show them all together. So we want to say when you can find no more than a single steady state. So these are the zeros of F, and here is the intersection with the parallel translate of this subspace S. Here says for any, in fact for any choice of rate constant, I don't know if it is said, there can be at most, if I didn't write it, it's a mistake. Something important in this statement is for any choice of rate constant. There is at most one positive solution, or we do the following. For any maximal minor, I didn't tell you which this is the name of the number of species, but for each maximal minor is there, but the cardinality d, this is the size of the rank of the matrices, D. You take all maximal minors, you take the maximal minor of A, the matrix A, which you put in a column, the monomials that give the parametrization. Okay. And this builds another matrix A. And you check whether these two signs, the determinant of this minor of A is the determinant of the same minor in the matrix S perp. And you see whether these essentially could be some zeros, but essentially they have the same sign all the way. Or you can go instead of S perp to S and instead of A to B. Or you can mix one of these with one of those, but if we took one on one side and one on the Gale dual side, there is a sign that comes out. But if all these signs are the same for any choice of maximal minor, then there cannot be more than one positive root, whatever the rate constants are. Okay? This is the flavor of the theorem. What did I do? Okay. So, so one question is, you know that there could be, that for some parameters and some total amounts, there could be more than one possible state. How do you find them? Okay. How do you get it? And as I said, Theorem 3 is based on all this previous work. And in this, as I said, we also give sufficient conditions for the hypothesis of this theorem to hold. And it's easy to implement this in we have a very basic implementation. In fact, there are fantastic implementations. I don't know exactly how they work. One is historical, is by Marty Feinberg and 6 students of him, which is still, it was made in DOS, Windows many years ago, but updated and it still works. You put it, tells you it is or it is not, and it gives you a choice of red constants and 2 different positive steady state for this choice. It doesn't give you which are the total amounts, but you compute the value of the linear functions defining S perp there, and you get the total amounts. And there are other packages that can be accessed from this wiki, Mathematics of Reaction Networks, and there are other things. And going back to this line here, once we decide that you have the capacity for multistationarity, we give an algorithm to produce vector of rate constant for which multistationarity occurs. And this is, as you can imagine, is based on the theorem of oriented matroids. Here, these are representable, but the theory tells you many things. And the main result, in fact, goes back to Rockefeller. He was doing convex analysis. And well, we have a non-professional implementation, but this can be easily implemented. Okay. So now I will just give you tell you a few words about the paper that should be already posted, but I am not editing it. That's my terrible mistake. It's something I've done with Frédéric Bihan. He's in France. Chambéry in France, and Magali Jaroli, she's a graduate student of mine. And we did something that is somehow related to what David told us about in this paper by Karin Gatterman and Wolfram. So what we took, Frédéric was visiting me in Buenos Aires, and he had just posted a paper, fantastic paper with Pierre-Jean Spelleauer, I don't know how to pronounce. But then there is a new version with the third co-author, which is Paco Santos. Is fantastic paper. So they try to find systems and understand how many positive roots you can find in different systems with combinatorial giving, and to describe many other things. It's a fantastic paper mixing real algebraic geometry with combinatorics. And so they had this idea that, so these points that are plotting there downstairs are the exponents that occur in my equations, in my polynomial equations. So you have the exponents in your polynomial equations, and you find what is called a regular, they use a triangulation, but it's enough to have what's called regular subdivision. This means that you pick your points. You lift your points for any point A, you have a lift, and you take the lower convex hull of this object upstairs and you project, and you get this decomposition downstairs. You're not going to pick all the points. For instance, this black point, which is in the middle of the yellow one, is not going to be picked. Okay? And it happens that we have this, it need not be a triangulation. And then they give a notion, what does it mean for one of these simplices to be decorated? So this is in the matrix of exponents, okay? But if you have a system of equation, you have exponents and you have coefficients of the system. So they give a condition on the coefficients of the system, which tells you when this coefficient matrix decorates one of these simplices. We just mean that if you look at the subsystem, which consists only of those, it has a positive solution. And then in fact, it is the implicit function theorem, or the standard, the yoga or Viro and all these tropical deformations. Then there's a way to define a deformation parameter, and for small values of this deformation parameter, our original system, if you can decorate 5 of these simplices, the original system will have at least 5 positive real roots close to there, except that it cannot be really implemented in a direct way. You have to do many things in order to be able to use this, because we have a system, it's not in the plane. We have a system with a number of variables goes to infinity, we cannot make pictures, and we cannot understand all this possible triangulation and subdivision. This is too much, but we managed, and we need to use the parameters. There are many issues, but this is the main idea. And then, for instance, let me show you, give you an idea of what kind of results we can get. So we know that this system has the capacity for multistationarity. So we want to describe rate constants, the case and t's for which this occurs. And this is the theorem that we get. This, so for this, with the previous notation, assume that the reaction rate constant satisfy this. What is there is the fact that the simplices are decorated, but you see they are it's related to some minors of the coefficient matrix. But you see that there is an open region that is defined in terms both of the case and the t's. Okay? They are involved, so if the rate of T1 over T2 is between these two quantities, then, and you also have to tune the other ones. This is explicit how you have to tune the other. So if gamma, the K5 and K4 have to be sufficiently big, you have to rescale them, and the quotient has to be big and one has to be big, so you need to. This is not as explicit. How big, you don't know, because it comes from implicit function theorem, but it tells you, well, if you make them sufficiently big and the quotient sufficiently small, then you will hit this region. So there are several other approaches to multistationarity, way many. The one that I love uses degree theory. And the last, the nicest one for me, this was already present in papers by Mincheva, and there's another paper by Mincheva and Carsten Conradi. And in this paper, it's more comprehensive. And they use degree theory. But the problem with everything that I am telling you, so I didn't tell you, but behind the scene, there is the Jacobian. And what happens is that this degree theory tells you is there are, or it's odd or even. It can tell you if it's even, you don't know whether it's 0, 2, or 100. If it is odd number, you know at least one. But it doesn't tell you how many, because it's an alternating sum of things. You just get the parity. So the problem is that in one variable, Rolle's theorem is a fantastic theorem. Rolle's theorem says between two roots of your polynomial, there is one root of the derivative. But you can turn this upside down. Is the number of positive roots of your polynomial is at most the number of roots of the derivative plus 1. And the derivative has degree 1 less, you can do induction. In several variables, it's completely unknown how to get there. There is a Chovansky-Rolle theorem that was made by Chovansky and improvement by Frank and Frederick. But we are far away, very far away from going from the Jacobian vanishes, and even if we find a number of points with this multiplicity, what can be said about the number of positive roots? Nothing that we know. So it's somehow easy and combinatorial to say at least one, but we cannot say how many. Okay? There are really, the math tools are not available. And it's a very difficult question. And so one way to circumvent this story is to go numerically. And there is nice papers. John is there in this paper with Heather Harrington and other people. And then also there is this other paper of Elizabeth Gross, Heather Harrington, and Smith Rosen, who was here. He was here. And Bern on the W and T pathway. And they go, essentially they use numerics to detect. So in theory, it's easy. You have to draw the discriminant in this incredibly high dimensional space, and you need to add something because you don't want this, each chamber in the complement of the discriminant, in each chamber the number of real roots is the same. You need to add some other hypersurfaces to say that the sign is not changing. And in one of these chambers, the number of positive real roots, the theory is there, but it cannot be implemented. It's terribly, the number of variables is so huge. Okay, so you can use numeric to try to find. Also, these guys in France, like Fabrice Rouillet and Mohab Safieldin, they have fantastic implementations to do this, to find a point in each connected component. But it's very complicated to describe this because there are too many. In theory, you can, but there are too many variables. And also, there also was mentioned there are several computations of small subnetworks, and then you can all extrapolations are implicit function theorem. So it happens very close to these, and you don't know how close, but there are many of those, and work on several of them. And then there is also some approaches in triangular forms that was proposed by Xiaoxian Tang, and is part of a paper that I hope we end soon. I'm also delaying the paper. And then the summary. Let me go the way. So summary is that we can use algebraic geometry to analyze system biology models. I would say maybe computational algebraic geometry, but there are also some general constants. And then this allows us to predict the dynamical behaviors of our model from the structure without simulations and without knowing the precise right constants. And in some interesting cases, I claim that we can see the woods and not only the trees. And as I said, in theory, you have several answers. In practice, they tend to be too complicated. And we need the use a combination of tools, not only from algebraic geometry, also from dynamical systems, numerical algebraic geometry, real algebraic geometry, biochemistry, etc. So as I told you, I try to use which variables are they put in a graph about, because they are two variables and for which you can see something. It's very complicated. It's really a jungle. And thank you very much. But let me end again, invited you to submit your best paper to this fantastic new journal. Thank you. Any question? Yes.
So in the previous slide, you said that sometimes in practice things are too complex. Is there hope? Like, will better computers help, or do you think it's better theory that's going to be what we need?
Standard computer or standard algorithms that we are using, I don't know.
I mean, this question about understanding the geography of parameter space, which is a description.
Yes.
This is really one. I mean, if there's time to think with the problem session, I'll talk about this. But those are inherently very complicated. The standard ways we have to look at it algorithmically make them even worse. And right, you have cylinder logic, algebraic decomposition, and so finding ways to understand them better, or study them, or navigate around them, or think about them in high-dimensional spaces instead of 2 dimensions, I think are really important open problems.
There are many, and then there are many open questions, very important. Some of the posters were about this, and understanding oscillations algebraically, or understanding stability, because these are real conditions. They hold in open, and it's complicated to work with a computer algebra, but it's way more complicated to work with inequalities. And so the theory is there. If you have a point, there's a root with conditions that are fantastic. You don't need to, but that tell you compute certain minors of a matrix, and according to the signs, you can ensure if all the roots of the polynomials will have negative real part, this will give stability or not. But then x, your point x is floating, is around in a huge dimensional space, and you can do it at one point. And all the books that you see are in 2D, Hopf bifurcation, whatever, all pictures, everything, all theories in 2D. In this number of variables, but I still think what we need to do, to say many things, that we need to adapt the theory and to find, which is meaningful, what can we do, what can we say. I find it very interesting. It's not enough to know the math tools that we know up to now.